Good afternoon to all the students. So yes, in the previous classes we have clearly learnt on the photosynthesis. Photosynthesis we have learnt in clear about non-cyclic photophosphorylation, and from dark reactions we have completely learnt the C3 cycle with bioenergetics, right? And in yesterday's class from respiration we have also learnt the process of glycolysis in detail. Today we are going to learn, just for a change, today we have allotted the practical session where we are going to learn demonstration of photosynthesis, that is starch test. Might be starch test is very basic, all the students have performed in the 10th standard, but today for a change we are going to learn a little different way. So here if you observe, this is the standard equation of photosynthesis, we all know, and as we have learned the discoveries also, once again let us recall. Who proposed that glucose is essential for the process of photosynthesis? Mole's half-life experiment. Water and sunlight are essential is said by Jan Ingenhousz. Glucose is the product of photosynthesis is proposed by von Sachs. Oxygen discovery by Joseph Priestley. Oxygen is coming from the process of photosynthesis as a byproduct is said by Jan Ingenhousz, and oxygen comes from water, not from carbon dioxide is said by C.B. Van Til. These are the basic discoveries we have done, and today we are going to prove that glucose is the main product of photosynthesis, and oxygen which is liberated is the byproduct. Right, ma? Glucose is the main product and oxygen is the byproduct. Where is the glucose formed? We have learnt in detail. If you have to take a plant, leaf, fine, right? Leaf is composed of what cells? Mesophyll cells. If I have to take a single mesophyll cell, in a single mesophyll cell around 40 to 60 chloroplasts are present. Fine, right? Around 40 to 60 chloroplasts are present. These organelles are present on the peripheral ends of the mesophyll cells. And if I have to take a single chloroplast, inside a chloroplast, which is a double membrane bound organelle, a third membrane system is present, which is known as thylakoid membrane. The membranous sac-like structure is known as thylakoid membrane, and the stalk of sac-like structures, is known as grana. The stalk is known as grana. Stalk of sac-like structure is grana. One grana is connected to the other grana by stroma lamellae. Fine, right? And the fluid filled space inside the chloroplast is known as what, ma? Stroma. Fluid filled space is known as stroma. We all know inside stroma glucose is formed, right? Glucose is formed where? Inside the stroma. The main product which is formed in stroma is glucose. This glucose, if it has to be stored as the food material in stroma, it will be stored in the form of starch. If it has to be transported to other cells for the process of respiration or for oxidation, it will be transported in the form of sucrose. Fine, right? So now in a leaf, whether starch is formed or not, okay, in a leaf is starch is present or not, we are going to analyze by starch test now. Got it, ma? So glucose will be stored in the form of starch, and now we are going to perform the starch test. For this, what are the requirements we need? We need ethanol, a beaker, test tube and leaf. A leaf which is placed, a plant which is placed in dark, okay, in dark for couple of days, and a plant which is present under the sunlight as usual. You need to pluck 2 leaves, one leaf from the dark plant, from the plant which is placed in dark for couple of days, and one leaf which is from the plant which is under the sunlight as usual. 2 leaves you have to take for this experiment. What you have to do initially is take the leaf, boil it in water, and then keep it in ethanol solution in the test tube, keep it in the beaker. Then you will get the leaf which has turned to pale in color like this, and the ethanol solution will completely turn to green. I have extracted that green color solution, okay, where chlorophyll will be completely absorbed by the ethanol, okay, and it turns to green color. Why we are performing the first test, that leaf is boiled in a water? Why is that? Because all the metabolic reactions that are happening inside the leaf, all the chemical reactions will be stopped, okay. The leaf processes will be arrested when it is boiled in the leaf, and later when leaf is kept in the ethanol solution, the chlorophyll which is present in the leaf is completely dissolved into the ethanol. Why chlorophyll? We have to remove the chlorophyll from the leaf, because when we are performing the iodine test, the chlorophyll which emits green color should not be dominant, okay. Chlorophyll has to be moved on, then only we can clearly analyze the test. And later after, you have to take that pale leaf from which the chlorophyll is removed, and one more leaf which is kept in the dark. Now these 2 leaves are ready for the iodine test, okay. Now this is the iodine solution which I have bought. From the dropper, carefully you have to take the iodine solution, and just few drops of iodine, keep it over the 2 leaves, drop it over the 2 leaves. Okay. So, here we are going to perform both the positive test and negative test of starch, positive test and negative test. Yeah, just place it for 1 minute or 2 minutes, and then you can clearly observe the blue black color of the leaf which is placed under sunlight, which shows positive test for starch, which proves that starch is formed at the end of photosynthesis. And one more leaf will turn pale yellow color, which is kept under dark. So, here we will get 2 tests. One is positive test and one is negative test. In positive test, if you have to see, the leaf turns to blue black color, the leaf turns to blue black color when it is added with iodine solution. And in the negative test, which is from the leaf which is placed in dark, here leaf turns to pale yellow color. Which shows absence of starch. Okay, leaf when added with iodine solution. These are the basic experiments, but then the effect of knowing these experiments, you will remember the concept for a long run. Okay, and the practical touch will make you the concept remember for a long run. This is starch test, positive. This is negative starch test. Got it right, ma? So, with this we have performed clearly, yeah, Here you can see two leaves, one where the veins are turning blue black color and the other turn to pale yellow, which is showing the absence of starch. So, with this our starch test is done, and in our next class we are going to perform plant growth and development.